So I'm very happy to talk to you today about the same topic that is actually also in the schedule. And I will also be presenting some of the results that of Klaus's studies. So today I want to talk to you about whether feeling blue makes you gray, or associations between depression and brain aging. So I'll go quickly, as we're a little overdue, but I think we all know that depression is highly prevalent, and it's also a problem for society. It's the leading cause of disability, and the global burden of disease is very high. And this is in part because it has a very high somatic comorbidity. So if you are diagnosed with depression, the risk of developing heart disease later on in your life almost doubles, the risk of diabetes, obesity, but also cognitive impairment is associated with depression, and that's also part of what I wanted to present here. So there are some pathways that are hypothesized how depression may affect biological aging, but also the risk of dementia. So one of these is that depression, at least in part of the patients, is associated with higher levels of inflammation and also increased cardiometabolic risk, which may lead to neural atrophy. But also unhealthy lifestyle behavior. So people with depression might be less likely to cook healthy food. They tend to smoke more, use more alcohol, and be less physically active, which are also factors known to affect brain health. And also cognitive inactivity, so less education, less continuous education or cognitive stimulation, less social interaction throughout life. So within our department, we've done several studies looking whether depression is associated with biological aging markers. I will highlight only a few here. So one of these is telomere length. So telomeres are on the caps of your chromosomes, and with the cell division, these telomeres shorten. So in a way, they give an indication of the sort of the biological age of a cell. And it is associated with age, and it tends to shorten with age. And so within this study in the NESDA cohort, so the Netherlands study for depression and anxiety, we observed that there were shorter telomeres in patients with depression, and it didn't really matter whether this depression was remitted at the time of the measurement or they had a current diagnosis. I don't know if there's so this is the baseline telomere length. In green, you see the control group, whereas in orange and blue, the people that had a depression either previously in their lives or at that time. And what was interesting to see is that we also have longitudinal data on these same individuals, and the rate is, so we see that the telomere shorten in 6-year time span. But this is the same decrease seen in all these groups. So this suggests that there's not a change in the rate, but it's really a stable difference, at least across the 6-year time span that we have in NESDA. So moving on to the brain, these are some findings from the ENIGMA consortium, which is also a large neuroimaging consortium, where they compared or pooled group studies looking at major depressive disorder compared to controls. And they observed that was lower cortical thickness in people with a depression, particularly in the medial prefrontal cortex. And in the subcortical regions, there was a slightly lower hippocampal volume. Although I must note that all these differences were rather small of effect. And in a brain age study where you really take the whole brain into account, so similar measures that Didac has also mentioned, it was observed that the patients with MDD had a slightly older brain age compared to the controls. But also here you can see that the distributions of the two groups largely overlap, and this accounted for about a year in older brain age in the depressed patients. So within NESDA, we also have longitudinal neuroimaging data. So this is work of Ilya Binovic. He is also part of Lifebrain and in the audience here. So within the NESDA sample, we investigated whether these regions that were associated with depression in the Enigma cohort, if these were also longitudinally associated with depression. We did not replicate the differences between groups in the hippocampal volume, but we did see differences in cortical thickness in the medial prefrontal cortex. So that was lower in depressed patients, but this was also stable across a 2-year follow-up and across a 9-year follow-up. And it was independent of changes in depressive symptoms across that time period. So I think that also signifies that these differences are stable over the time period that we measured. So suggesting that there is no acceleration of biological aging, at least not in the adult lifespan that we investigated here. So this biological aging markers, so the telomere length, but also cortical thickness, was not perplexively related to depression course, which suggests that it might be something that happens either early on in the disease or it may be a predisposition to the disease. And I think that fits really nicely with other things that have been said throughout this day, really. So what do we look at in relation to depression in brain in Lifebrain? So one of the things that we wanted to see was whether these associations between depression and brain structure were present across the lifespan. So previous work, mainly in Enigma, usually had only limited age span, so up to 65, most of the cohorts. So within Lifebrain, we have a much larger age span, so we were interested to see that this was also present in the elderly and in the general population, so also in subclinical levels of depression. And we took the same regions that were investigated before, so the medial prefrontal cortex and the hippocampus. And we meta-analyzed, or Julia meta-analyzed, I must say, data from 6 population-based cohorts within Lifebrain and 2 patient control cohorts within Lifebrain. And we did replicate the findings also seen in Enigma, was that regional gray matter was reduced in these regions in people with depression, but this was only present in the clinically depressed population. So we also looked at the severity of depression symptoms, so in a more dimensional way, and this was not associated with regional gray matter in the population-based samples. And it was also consistent across the adult lifespan. So if we looked at young adults or mid-age adults, there were no differences in the associations. Although I must say that we did not have many clinically depressed elderly, so that may be something that still needs to be investigated by other studies. Then I shortly want to go into late onset depression. So this is work by Klaus's group and also Didak. So late onset depression might be somewhat of a different type of depression than early or middle-age onset depression. So with middle onset or early onset depression, the idea is that depression plays a causal role, whether directly or indirectly on these brain differences. But in late onset depression, that is thought to be the other way around. So that things are going on in the brain, such as hypertension that damage the brain, that leads to depressive symptoms in later life. So a hypothesis by Klaus and his colleagues was that late onset depression would be then associated with larger global vascular atrophy and not so much with specific regions, but also that this would be associated with a faster rate of atrophy. Compared to people with an early onset of depression. So they investigated this as preliminary data in 6 Lifebrain cohorts so far, where they also had longitudinal data. Hypothesis 2 is something that you can ignore, but they did observe evidence for indeed global atrophy in people with late onset depression relative to early onset depression. And you can see it in this graph. So in green, there is ventricle size in the late onset depression. And here on the x-axis, you see age. In blue are early onset and the controls. So you don't see much difference in the ventricle size over age between the early onset depression and the controls. But in the late onset depression, you see that there's an increase or a larger ventricle size, and that increases over time, not only with age, but also the rate is faster. So to conclude, does feeling blue make you gray? I think depression is associated with various markers of biological aging, but we don't see very consistent evidence that this aging is going faster in people with depression, with the exception of late onset depression, which may be a different thing. So I think it's still important to prevent depression for obvious reasons, but perhaps also for healthy brain aging. But I think the most important open question here is, and that's something that maybe we will hopefully also be able to address it within Lifebrain, is when does this aging occur? And with that, I want to conclude. Thank you.
Thank you so much, Sarah, for a great talk. Sorry that I was a little surprised by the switch between you and what is great with the entire the group and the NESDA, but it focuses on mental health, which is also a great focus of Lifebrain, even though we have not, well, many presentations are on function. So I think that these were really interesting data. I have some questions, but I think now is the time when we have come to the discussion for this entire session. So you may ask questions to all of the speakers. So maybe you guys will come down here. So I saw that Anders raised his hand first, and then Ulman, and then David. And I don't think, yeah. So.
Yeah, thanks for all the presentations. They were great. I have a question for you, Alvaro. Your brain stimulation studies are intriguing and very interesting, so thank you for that. Do you believe that the effects you show will last for a long time, or do you think these are kind of short-term symptom effects?
That's a good question.
Alvaro, will you please either take a mic?
Thank you. Yeah. Sorry about that. I think it's a great question. We don't know. I mean, we can speculate, but we don't know. The studies, as I showed, were largely proof of principle little studies, tantalizing, I think, but had a follow-up generally of 3 months. And I think what we know from brain stimulation treatments and approaches in general is that it's unlikely to lead to lasting or everlasting changes, but rather need some sort of maintenance. Whether by repeated application you can induce longer lasting effects, that's I think still an open question. Part of the reason why we were interested in this personalized home-based application is because with home-based, it's fairly straightforward to do a maintenance. And so we're doing that in the Alzheimer's data where we have the longest follow-up. We now have people that have gone on maintenance for 2 years and are maintaining the benefit, which is a very small number, and it's hard to make too much out of it, but at least it's safe, it can be done. And so, yeah, I think.
Thank you. So, Ulman.
I have a question to Laura. So we know that depression is defined by a series of symptoms. And of course, the etiology may be different for different kinds of depression. And in that context, I found your diverging findings for late onset depression quite interesting. And it was tempting to think of whether late onset depression may actually be a reaction to you noticing your brain falling apart. And I just wonder whether there's anything that could be used longitudinal data to find out whether late onset depression is a different story, so to say, has a different story to tell from depression as we typically know it. I mean, then probably the genetic predictors should also be different and so on. So I'm just curious what you think.
I think it's a very relevant point, and also something that we really focus on within our department is to look at the heterogeneity within depression. So, like I said, the inflammation, for example, metabolic effects or remission in subgroups of people with depression.
When it concerns late onset depression, maybe it'd be nice to hear from Klaus, as I'm not a big expert in that respect.
It depends by what you mean by reaction. I'm disinclined to think it's a, let's say, a psychological understandable reaction. My guess is that it's a result of the same processes that a little bit later lead to dementia.
Yeah, and I think my question was whether it's kind of a direct common cause or whether there is some appraisal loop in it, in the sense that you just notice things about yourself that make you feel sad.
Yeah, we've done this only in Whitehall, and there late onset is associated with vascular risk. Not the others. It's not associated with immune markers, which would have been another theory, although, you know, they are, I think the error, the measurement error there is really quite great. But I think they're both associated with vascular risk upstream and cognitive impairment downstream.
I won't go before David, but just a comment, Laura. I was also thinking about this late onset depression and whether it is a symptom of somatic disease, because it can, for instance, be a symptom years before Parkinson diagnosis because your dopamine levels are falling and you feel depressed. Have you been characterizing them in terms of just gross health?
So not in terms of the late onset depression. We did look at various biological aging markers, so epigenetic clocks, proteomic clocks, those kind of things, and associating them with all kinds of health factors. There we did see, of course, strong associations with BMI, but also with general somatic health and number of chronic diseases. So it's not exactly what you're going into. Because within NESDA, we're limited until the age of 65. So yeah, maybe in Whitehall, I don't know.
Okay, thank you. Very, very interesting. So, it was David.
Yeah, thank you. I have a couple of questions for Laura and Nayara, very short ones. For Laura, I actually had the opposite question. What about child onset depression? I mean, can we say that then if there is an early onset depression and a midlife onset depression, it is quite, I would say, the same? It's something that is there, doesn't change or develops in parallel over lifespan, then late onset depression may be different. So what is the earliest age that we know if these changes in hippocampus and ventromedial cortex are also evident in child with depression? Or because the data that you send, the data that you show, it only, I think it's only 20 years, the lower range, right? Something like this. So it's something that we would expect similar brain changes in children. Do we know that? I'm curious about that.
Yeah, so within Lifebrain, we decided not to include the child cohorts because there was very limited information on depression. I do think that the early onset depression, or there are some findings suggesting that it might be a different type of depression also compared to adult onset, midlife onset. So I think this is really relevant, and also to study this longitudinally in children. I'm not aware of studies, but I will look into it.
Okay. As a curiosity. And Nayara, for your study, you didn't find any, if I understood well, any kind of interaction between so the prediction is the baseline physical activity predicts the physical activity performance over the 4 years follow-up, or the follow-up that you had in these 3 different groups is the main predictor, the baseline physical activity.
Yes.
But then you didn't find any kind of interaction with the intervention, even if the distribution was equal as you showed. Wasn't there any kind of statistical sort of interaction, sort of thing that for those that have lower or higher baseline may work more or less?
So you mean an interaction between the group that they were assigned with their physical function and further? We actually haven't looked at interactions. We didn't include we looked at interactions, but not using the randomized control group. So we sort of got to the point where we actually went the other way, and we thought actually the randomized control group isn't telling us that much more at year 4. There have been some follow-up from our group where they have done questionnaires at year 2 and asked, from each group, how much of these physical activities do you still follow up on, and how much are you still doing? And at year 2, I think it was something that 24% of anyone that had been randomized to one of the muscle strengthening activities were continuing with any form of what they had been instructed to do. And what we did see is that people in the control group actually, and you see this in several interventions, is people in the control group get a bit more conscientious and think about what physical activity they should be doing and might pick up some other behaviors.
Are there more questions? Yes, please.
I have a question for Professor Pascual. You mentioned a little bit that you are thinking of maybe using some more sort of naturalistic data from mobile devices in looking at assessment and early detection. And I was wondering if you have used that already, or you're just thinking about it, and whether you think that that's worthwhile doing, or actually having a test on an app is good enough, and then we should just stick to that because it's better control and we have more.
We're using them. I don't think we have a clear answer as to whether it's better or not in the app. I think that one of the problems with the app, at least in my experience, is that the adherence tends to not be very good. So you need to design real repeated pings, as it were, to coach people into staying with it, and the attrition rate tends to be quite high. So I think that is true in a study, in a population scalability effort. It would be a very challenging thing, and so I think the more passive we can capture some of these measures, the more value it's going to have. And so we're doing the studies to try to figure that out. The little sort of adaptation of the dual task cost is one such example. It stands to be seen. To address the point you were saying as to what my gut feeling is, I do believe that that's going to make a big difference, but we'll find out.
Thank you. I can see that Lars Nyberg has raised. You have raised your hand, Lars.
Yeah, to prevent you from having coffee. Since I'm not getting any here. And Nayara, I was wondering about your findings about sort of the role of physical status at onset. And I think it resembles data on sort of when people sign up for the military. You can see that sort of their fitness level, grip strength, cardiovascular fitness at that stage, say being around age 18, is quite predictive of sort of later life events. How do you see that sort of physical status in relation to genes? Is it some sort of a genetic predisposition, or is it really sort of, should we really exercise our kids?
That was a great question. I don't know how much of a role there is. I'm sure there must be some influence of it, but I don't know, and I haven't looked at that before. What I would say is that this earlier set it looks like it's earlier and earlier. So for example, there was a study that came from Rick's group where they showed that actually midlife physical activity or physical function was a much better predictor than the late life for their, I think it was cognitive outcomes. And that seems to be the case that we find as well. So it does, I don't want to say that it doesn't matter. Of course, if you can change the behavior and you can improve, there must be something good to that as well. But it does look like these earlier functions have a very big significance. Now, whether that is coming from birth or from very early lifestyle, it's hard to say. I would add as well that it's very rare that you see people that have a complete lifestyle change. So when you ask people about what their behaviors were like early on, they tend to have had these tendencies from earlier on as well. So it's very hard to disintegrate these things.
I would also add the LISA cohort is unique in that it's very, very active. So it is unusually active. I think their average steps per day was something like 9,000 steps, which isn't necessarily representative of any of the other cohorts we've worked with.
Great, thanks.
There was a hand up.
Yeah, and I also see Christian had a hand raised, and you had a hand raised. We will go a little into the coffee break if that's okay.
No, that's fine. We can make it up later. No
Yes,
problem.
okay. Yeah, so then we do that. I think I saw Christian first, but you know who went first, you tell me. I saw Christian first, so then Christian.
So just a short comment to the childhood, the early training of muscles. A guy in Oslo, Christian Gundersen, has used a mouse model to train mice in different stages of life. And he can show that you can change the number of muscle cells depending on when you train the mice. So, and that is pretty convincing data. So I think that in principle, at least you can do it by training certain periods of young life.
That would suggest that the younger ones are.
Yeah.
You also get the metabolic steroids, didn't they?
No.
I don't get it. The mice.
He in one of the studies he probably did, but in the other ones he just trained them.
Yeah. Okay.
Yeah.
So maybe not the steroid part.
Okay, we have a next question. Please.
Just very quickly, it's kind of to link to Naira's talk. Isn't it slightly that we are doing a little bit too little and a bit too late? Because I come from chronic pain, physical activity, and now small vessel disease. And by the age of 65, so when you are starting the physical intervention, over half of those people will already have brain changes caused by their unhealthy lifestyle. And you said they do 9,000 steps. With people from physical activity, we try to get people run 10K in their 60s, which is way beyond 9,000 steps. So maybe those are the changes we should be doing, because I looked at UK Biobank and midlife changes predicting what will happen when we started scanning their brains when they're in their 70s. And it's already, people are on completely different trajectories. And to make this change, you should have started when they're in the 50s. I know that 50 is now the new 30, but maybe when we start seeing those people in the clinic, because my patients that we see are in their late 60s and 70s. For many of them, they're still on this quite good trajectory. For many of them, it's just too late. You cannot get them to move or to turn their life around. So, yeah.
So there are two things I'd like to comment there. So I do agree that I think there's a huge benefit to trying to promote these healthier behaviors much earlier. And that fits in with what we're saying before. It might actually be, much, much earlier that we should be focusing on. But I don't think that I think there's still a lot to be gained by having interventions in older adults, because it goes beyond these, there are other benefits that we reap that also have an effect on cognition and on mental health and on well-being. What we saw in REACT is improving mobility also has a big, cascade of effects on functional independence, on improved balance. And all of these also have, their own effects. So I think there's still a lot of benefits there. And I should clarify that the 9,000 steps, that was independent of intervention. So at baseline, they were already, that was just where they were. So it is very high. And of course, that's led to people to question whether having a cohort who's that physically fit, can we actually really still see those advantages? Should we be focusing on the ones that are more sedentary? But groups have also done that. So there's groups in the States, also in REACT, actually, the inclusion criteria was that they were at risk of mobility impairment. So they were at completely a different end. I don't know how many steps they did a day, but I would have guessed it was a lot less. But that actually didn't change. We had this maintenance effect in hippocampal volume, but we didn't have huge effects overall when we didn't have any effects on cognition from that physical activity intervention.
Are there more questions?
Then, with the risk of having my head chopped off, I have a question for Rick because we're into the coffee break. But I just wanted, so first, Rick, I think it's great that you focus on change. We weren't able to do that in the overall Lifebrain cohort because there are so different measures across times for general cognitive ability. But Rick looked at that, and I think it's interesting. You found this with area, and in this regard, I didn't get to think about the plot fully. But so area changes with age, but it changes much less than thickness, right? I would think. So I would think that the variability you have in area change is lesser. So would you care to comment why do you think it is that you find this with area specifically? Have you looked at the variability in the metrics?
I can't remember the data. I can look at it after this talk to see whether you're right. You might well be right that the change in area is much smaller than change in thickness. But I would have thought that in our latent change score models, when we do a test of the Steiger test to change there is a difference in the slopes. That should, I would have thought, but correct me if I'm wrong, allow for the fact that one might be noisier than the other. But it's certainly, yes, if it were just a lack of significant correlation in one and a significant in the other, it could just be a range effect. But the fact that
I'm not saying it is. On the contrary, I think I don't think it is. It's just I was just trying to get at it because I would think that the variability in area changes.
I suspect you're right, and I'll go and look at the data. Yeah.
But yeah, let's look at that. So okay, thank you. Then I want to thank all the speakers for great talks and interesting discussions, and also thank all of those who asked questions. And now we will have a coffee break, and we will extend it because Barbara promised that. So a whole 20 minutes coffee break now. Okay, thank you.